Hello, welcome back to Unit 2, Session 5 of this course. Under this session, we'll be looking at the first part on the movement of substances in and out of the cell. At the end of this session, you should be able to explain partial permeability and also name two types of protein present in the plasma membrane and define active transport, and also describe what happens when a plant cell is placed in a hypertonic solution, and also state two differences between channel and carrier proteins, and then finally explain how cystinuria develops. The plasma membrane functions as a boundary that separates the cell from its surroundings, and it allows the movement of certain substances through it whilst restraining others. And the component molecules of the plasma membrane helps in this selectivity process possible. And so a steady traffic of small molecules and ions are able to move across the plasma membrane in both directions. That is, these small molecules and ions could move from the cytoplasm of the cell to the surrounding of the cell, whereas these molecules and ions could also move from outside the cell across the plasma membrane into the cell's cytoplasm. Two main types of proteins are found in the plasma membrane, and these are channel proteins and carrier proteins. So the selective permeability property of the plasma membrane will depend on the discriminating barrier of the lipid bilayer and the specific transport protein that is found in the membrane. And so in order to understand the direction or flow of substances across the plasma membrane, we should be able to know the mechanism that actually drives these molecules across the membrane. And so in the next slide, we'll be looking at the mechanisms that actually drive molecules across the plasma membrane. And the first one that we'll be looking at is the active transport. So in passive transport, there is movement of substances down a gradient across the membrane, and this does not require the use of energy, or no energy is involved during the movement of the substances. An example is diffusion, osmosis, and facilitated diffusion. And there are factors that determine passive transport, and these are concentration gradient, temperature, the size of the molecule, and the medium permeability. The first example of passive transport we'll be looking at is diffusion. And diffusion is the movement of particles of a substance from a highly concentrated region to a region of lower concentration. And it will occur until the particles are evenly distributed. So any substance will diffuse down its concentration gradient, and the concentration gradient is the region along which the density of the chemical substance decreases. So the gradient represents the potential energy, and this potential energy drives the process. Is the potential energy that drives the diffusion process. A diffusion is a passive or spontaneous process, and it does not require the use of energy. So for instance, in cellular respiration, it requires the use of oxygen, and this oxygen usage for cellular respiration occurs by diffusion. So as the respiratory process continues in the cell, the oxygen will continuously be consumed. And another example is in amoeba or in single-celled organisms, whereby there is an exchange of gases across the general body of the single-celled organism, and this exchange of gases occurs by diffusion. Now let's look at osmosis as a type of passive transport. And osmosis is the movement of water molecules from a region of lower solute concentration to a higher solute concentrated region across a semi-permeable membrane. And what happens in osmosis is that this process will continue until both solutions attain the same concentration. So looking at figure 8, which is a representative diagram of osmosis, it indicates that the solution on the left side has different concentrations, which is divided with a semi-permeable membrane. So there is the movement of water from the region whereby there is lower solute concentration. So water moves from the lower solute concentrated region to the higher solute concentrated region, and that's seen in the second beaker, whereby they've all attain the solution in between the semi-permeable membrane, or the solution that is found on each side of the semi-permeable membrane have attained the same concentration gradient. So in the course of movement of water to attain the same concentration on each side of the solution, there is the buildup of pressure, and this pressure that develops when the solution takes up water by osmosis is called osmotic pressure. Let's have a look at the effect of osmosis on living organisms. Now, the movement of water across the plasma membrane and the balance of water between the cell and its environment have important consequences for living organisms. The ability of a solution to cause a cell to gain or lose water is called tonicity, and it takes into account the solute concentration and membrane permeability. So when a cell is placed in a hypertonic solution, for example, an animal cell that lacks cell wall, the cell will tend to lose more water, or water will leave the cell into the solution, and by so doing the cell will become, or the cell will shrink and therefore could die as a result. And when such a cell is also placed in isotonic solution, this isotonic solution has the same concentration as the inside of the cell, and by so doing the rate at which water moves into the cell will be the same as the rate at which water moves outside the cell. And the other example is the hypotonic solution. When that same kind of cell is placed in hypotonic solution, the effect is that the water will move into the cell, and this will cause the cell to swell up and it could die eventually. So that's the case for most terrestrial animals, and because the cells lack cell wall, they can neither tolerate excessive uptake or excessive loss of water, and it is so ideal in that when they are placed in isotonic solution. Now have a look at this question. Can you explain the terms isotonic, hypertonic, and hypotonic solutions? Hope you were able to do it. Animals and other organisms without cell wall require special adaptations for regulation of water levels in the cell. An example being Paramecium, which lives in fresh water, which is hypotonic to the cell, has contractile vacuole, which pumps excess water out of the cell. Cell walls of plants, prokaryotes, fungi, and some protists helps to maintain the cell's water balance. And when such a cell is placed in isotonic solution, there is no net movement of water particles, and the cell membrane is attached to the cell. However, in hypertonic solution, the water particles move out of the cell. The cell membrane rather shrinks and detaches from the cell wall, and this phenomenon is referred to as plasmolysis. On the other hand, in hypotonic solution, the water particles move into the cell, and the cell wall counteracts osmotic pressure to prevent swelling and lysis. Now let's have a look at facilitated diffusion as example of passive transport. So this facilitated diffusion involves the movement of polar molecules across the plasma membrane that requires the help of transport proteins embedded in the plasma membrane. And two types are known, that is channel proteins and carrier proteins. Starting with channel proteins, they function by having hydrophilic pathways through which water molecules or small ions are able to cross the membrane. An example is aquaporins, and these are found in the membrane of cells, and it enhances the rate of water movement across the plasma membrane. Apart from aquaporins, there are also ion channels, and these gated channels or ion channels are responsible for nerve impulse transmission through sodium-potassium pump. And now let's have a look at the carrier proteins. They are transport proteins and they hold on onto their passengers and change the shape in a way that helps in moving the substance or the polar substance across the plasma membrane. Now let's have a look at cystinuria. And this is a condition in humans and it develops when carrier proteins that transport cysteine and some other amino acids across the plasma membrane of the kidney cells is absent. And by so doing, the individual with this condition develops painful stones from amino acids that accumulate and crystallize in the kidney. So under Unit 2, Section 5 of this course, we've studied the selective permeability of the cell membrane, and we also got to know the nonpolar substances cross the plasma membrane by dissolving in the lipid bilayer, whereas the polar substances do so through transport proteins. Both osmosis and diffusion are passive processes because substances move down concentration gradient. And also the effect of osmosis on living organisms are also considered. And also look at the final one, cystinuria, which is a human condition whereby carrier proteins that transport cysteine and other amino acids are absent, resulting in the formation of kidney stones.